My name is Dr. Aruna Diwan and today's topic is meiosis or meiotic cell division. Before starting the proper stages of meiosis, let us understand few concepts first. The first one is homologous chromosomes. Homologous chromosomes actually play a very important role in meiotic cell division. So it is important for us to understand what are homologous chromosomes and what are non-homologous chromosomes. The homologous chromosomes are actually a pair of chromosomes of which one chromosome is from mother and the other one is from father. Let us understand this with the help of a diagram. Let us suppose this is a diploid cell. We know that in a diploid cell we have 23 pairs of chromosomes. Let us suppose this is first pair of chromosome, this is second, this is third and so on. This is 22nd pair of chromosome and we know that in 23rd, in case of female, one will be X, other one will be, both of them will be X, and in case of male, one will be X and other one will be Y. Now, this pair is a homologous pair. Now, how can we say that this is a homologous pair? For that, we need to understand few characteristics of homologous chromosomes. The first one is that the length of the homologous pair of chromosomes is approximately same. And second one is the position of centromere. The position of centromere of homologous pair is always same. According to the position of centromere, we know that chromosomes are divided into four types: metacentric chromosome, submetacentric chromosome, acrocentric chromosome and telocentric chromosome. Now what is metacentric chromosome? In case of metacentric chromosome, the position of the centromere is in middle of the total length of the chromosome, right? So it will divide the chromosome, the two arms of chromosome into equal lengths. The second one is submetacentric chromosome, in which in case of chromosome the centromere will be slightly away from the center. So the arms will be divided into two slightly unequal lengths. In case of acrocentric chromosome, the position of centromere will be near to any one end of the chromosome. So the length of the two arms are very unequal. And in case of telocentric chromosome, the position of centromere is at any one end of the chromosome. Now we were talking about the homologous chromosomes. The third characteristic of homologous chromosome is the position of genes or the locus of genes. For that, let us make a homologous pair of chromosome a little big. Let us suppose this is a chromosome of mother and this is chromosome of father. Right? Here you can notice that I have drawn the position of centromere similar in both cases. Now we were talking about the third characteristic of homologous chromosome. In this case, the locus of gene will remain same. For example, if this is the position of a gene of a mother's ear shape, then at the same position the gene of ear shape or shape of the ear in this chromosome will be same. So these are the total three characteristics that shows us that which one is the homologous pair and which one is not. Now in this case we cannot say this and this are homologous pairs. Why? Because we can clearly see the length is different. So this is the concept of homologous pair of chromosomes. Now let us move to the next concept, that is what is the difference between small n and capital N. Difference between small n, as we say, if a cell is diploid, we say it's 2n, right? Then what will be the number of n in that cell? For that, let us draw again a diploid cell in which one of the chromosome is maternal and other one is paternal. Right? The small n is actually the number of centromeres present. Here how many centromeres are present? In this, 1 centromere means it's 1n, and in this again 1 centromere means it's 1n. So total it will be 2n. Right? Now again, in case of n, it will be the total number of DNA present. Now here how many DNAs are present? As this is still not replicated, so this is having 1 DNA and this will also have 1 DNA. So this will be having 1n, and here we can write 1n for this also. So here we have 2n means diploid number of cell, and 2n means 2 DNA are present. Now in case of interphase, we know that in interphase, in S phase of interphase, the DNA gets replicated. Let us draw the replicated DNA now, replicated chromosome. Here, when this gets replicated, it will be like this, and when this will get replicated, it will look like this. Now in this case, if we talk about small n, then how many centromeres are here? Centromere is still 1, so this will be 1n. How many centromeres are here? Centromere is still 1, so this will be 1n. So total it is 2n, again diploid. Now in case of replication of DNA, the DNA has doubled. So how many DNA are here? Here 2 DNA are there, so it will be 2n. And again here 2 DNA are there, but centromere is still 1. So this will be 2n. So total will be 4n. Now in this case, it is a single structured chromosome. Both are single structured chromosome. So this will be a diploid cell, but the DNA will be 2n. And here in this case, these are double structured chromosome, but still they have one single centromere. So it will be a diploid cell, but they will be having 4 number of DNA. So it will be 4 capital N. Right? I hope this concept is clear. Now we can move on to the meiotic cell division. We know that in mitosis, the daughter cell that are formed are identical to the parent cell in case of number of chromosomes. But in meiosis, this number of chromosomes get reduced. Why? Let us understand this. Let us suppose this is a cell having 4 total number of chromosomes. Now in case of mitosis, what will happen? This will form 2 daughter cells, and both daughter cells will be having 4 and 4 number of chromosomes. Right? Again, when they will divide into 2 daughter cells, again they will be having 4 and 4 number of chromosomes as per the method of mitotic division. So now there is no problem. But in case of meiosis, we know that in case of meiosis, the 2 gametes unite. This is okay with normal growth of the organism as well as for asexual reproduction. But in case of sexual reproduction, meiosis is compulsory. Why? Because in case of sexual reproduction, now what is going to happen? 2 gametes will fuse together to form a zygote. Let us suppose this is a cell of having 4 number of, this is a gamete having 4 number of chromosomes. This is another gamete having 4 number of chromosomes. When these 2 gametes will unite, then there will be 8 number of chromosomes. Now when this will when this will again unite with the another gamete in next generation, then they will end up with 16 number of chromosomes. Now this is a difficult situation, right? This is not going to happen. So there must be some solution for this. What is the solution for it? The solution is that the gametes, when the gametes are formed, the number of chromosomes are reduced. Let us understand that situation also. If gamete is having, if a somatic cell of certain organism is having 4 number of chromosomes. But for that organism, if their gametes are having 2 number of chromosomes, 2 gametes, 2 number of chromosomes, then they will unite to form 4 number of chromosomes. The organism having 4 number of chromosomes, so it will be same as its somatic cell or parent cell, right? So this is the situation where meiotic cell division is required, in which the cell division will reduce the number of chromosomes into half while forming gametes. Now let us move to the actual meiotic cell division. Meiosis is actually divided into 2 parts, Meiosis One and Meiosis Two. In case of Meiosis One, the division is reduction. So Meiosis One is reductional division, right? And Meiosis Two, in case of Meiosis Two, it is exactly like mitosis. So it is equational division. Okay, Meiosis One is again divided into 4 phases: Prophase One, Metaphase One, Anaphase One, and Telophase One. Similarly, Meiosis Two is divided into 4 phases: Prophase Two, Metaphase Two, Anaphase Two, and Telophase Two. Right? Now, first we will start with Meiosis One. In Meiosis One, we have Prophase One. Prophase One is the longest and most complex phase of Meiosis One, right? So for sake of simplicity, it is again divided into 5 substages. The first one is lactotine, then zygote, pachytene, diplotene, and dikinesis. They are also known as lactonema, zygonema, pachynyma, and diplonema, right? Let us start with lactotene. Lactotene is the first stage of Prophase One. At this stage, we know that the first and foremost thing will be interphase. Interphase will happen and then it will move to the meiotic cell division. Now I am not going to explain the interphase today because that I have already explained in my previous video, that is mitotic cell division and its checkpoints. It is explained in very detail in that video. Okay, so I am not going to explain interphase now. Let us directly jump to Prophase One, or let us directly jump to meiotic cell division. Now, in lactotene stage, as we know, in the S phase of interphase, these chromosomes are replicated, the DNA are replicated. As the DNA are freshly replicated, so what will happen? Those DNA, the chromosomes will look like a long thread-like structures. In lactotene stage, chromosomes will be like long thread-like structures. But in these long thread-like structures, there are few bead-like structures that can be seen. What are these bead-like structures? These bead-like structures are chromomeres. They are also known as idiomeres. What are these? These are actually the highly coiled structure of DNA and protein complex. At what place? At the place where transcription is absent. So we can see a long thread-like structures in which few beads can be seen. Bead-like structures can be seen. Okay. Next is zygote. Now in case of zygote, what will happen? This is very important stage. Here what will happen? Here comes the homologous pair. Now in case of mitosis, as we know, there are 46 chromosomes, or 23 pairs we say, but 46 chromosomes are randomly arranged. But in case of meiosis, the homologous chromosome will start looking for its pair. Means if this is a maternal chromosome number 1, then it will start looking for the paternal chromosome number 1. Let us suppose this is a paternal chromosome number 1 lying here. Okay. This paternal chromosome is lying on the opposite side. But this maternal chromosome will start looking for paternal chromosome and they both will come together. This paternal chromosome will arranged near maternal chromosome, right? In case of interphase, in S phase of interphase, these are already duplicated. So we will draw them like this. This is maternal chromosome and this is paternal chromosome. Now they are paired, right? Now as they are paired, they are said to be homologous chromosomes, and this pairing is known as synapsis. The synapsis actually occurs in a zipper-like fashion. It can start from any location, but it will be in a zipper-like fashion. So this pairing is known as the both of the length of the both will be same. They both should look similar, right? Now they are arranged in pairs in case of zygote. Now next comes the Pachytene. What will happen in Pachytene? In Pachytene stage, these two, here they will look double structured, right? Now in case of Pachytene stage, what will going to happen? These are, you know that these are two sister chromatids, okay, of maternal chromosome. This red one is maternal chromosome and this black one is paternal chromosome. And these two are sister chromatids of paternal chromosome. Okay. Now they both will start crossing over. In case of Pachytene, these both will start crossing over, and crossing over will happen where? Crossing over will happen with one sister chromatid of this with the one sister chromatid of paternal chromosome. It will look like this. Okay, they have started crossing over. This crossing over will happen in Pachytene stage. This is the most important stage for studies of meiosis. Why? Because at this stage, first of all, these chromosomes have started shortening or condensing and thickening. So at this stage, chromosomes are very clear for their studies. Even they are more clear than the chromosomes are found in metaphase of mitotic cell division. Okay, so in case of Pachytene, what will happen in case of Pachytene? These will start crossing over. Okay, now let us move on to the next stage, that is diplotene. In case of diplotene, what will happen? In case of diplotene, these both will start moving away from each other, right? These both will start moving away from each other. Now what we can see? Now we can see very clearly the places where they are connected or where they are joined. These places are known as chiasmata. These places are known as chiasmata. Now let us move to the last stage of this prophase one, that is diakinesis. There is not much difference between diakinesis and diplotene. The only difference is that in case of diakinesis, the chromosomes still get more condensed and more thickened, and more clearly we can see the chiasmata. Okay. Now let us move to metaphase one. We know that in case of metaphase one, these chromosomes starts arranging at the equatorial plate, right? So this will arrange at the equatorial plate. It is like this, right? Suppose there are two, then these two will arrange at equatorial plate. Let us suppose this is the another one chromosome pair. These have arranged themselves in the equatorial plate. Simultaneously, in the prophase one, we know that the nuclear membrane gets disappeared. These all things are already explained in mitosis. So if you have any doubt, you can watch that video. In prophase one, the nuclear membrane also gets disappeared, as well as the spindle starts forming, right? These microtubules start attaching to the chromosomes at the centromere of chromosomes. Now, in case of mitosis, what we have seen that when anaphase is arriving, then in case of mitosis, what happens? The centromere starts splitting from longitudinally from middle. But in meiosis, the difference is that here centromere will not split. Only these two chromosomes will move away to the opposite poles. But the thing is, now the maternal chromosome is not fully maternal and paternal chromosome is not fully paternal. What will happen here? Crossing over was happened. So this part is a paternal chromosome. Again here this part is of paternal chromosome. And in this side, when anaphase will happen, when they both will move on the opposite side, then it will look like this. Okay, so crossing over has done. They have exchanged their material, right? Now, as they have exchanged their material, now they are in anaphase stage. Then they will move to telophase, and this will form two cells. After cytokinesis, in some cases cytokinesis is followed after every meiosis, after meiosis 1, as well as it will be followed after meiosis 2. But in some cases cytokinesis is postponed and it is directly done in after meiosis 2. But here we are showing the cytokinesis. First telophase will happen. In case of telophase, you know that the nuclear membrane will start appear again, and both the chromosomes, they will be lying inside. Now this maternal chromosome is having few parts of paternal chromosome, and this paternal chromosome, they are having few parts of maternal chromosome. Again I have drawn only single chromosome just for the sake of simplicity. So this is after cytokinesis, the telophase is done. After telophase, we know that it will start furrowing, the cell will start dividing into two. This is the stage after cytokinesis. We have two daughter cells now. Okay, in this we had these two chromosomes together in one cell. Now we have one chromosome here and one here. So this is a reductional division, and this is now N and N. Now it will move to the meiosis 2. In meiosis 2 will look like exactly like it will be, the process will be like mitosis. What is going to happen? These are the stages. Prophase 2, Metaphase 2, Anaphase 2 and Telophase 2. Okay, what will happen in Prophase 2? In Prophase 2, now we are having, we are considering only one of the cell. This one of the cell is of maternal chromosome. The similar stages will move with the paternal chromosome cell. We had two daughter cells there. Again here we are considering one daughter cell. Now what will happen? Now this is prophase. In case of prophase, again nuclear membrane will get dissolved and it will move on to the metaphase, where this will arrange on equatorial plane. This will get arranged on equatorial plane. Same that was done in mitosis. Okay, as this has arranged in equatorial plane, the spindle fibers will get attached to this. Now in Anaphase 2, what is going to happen? This will get split from the middle, and half of the chromosome will be here and half of the chromosome will be pulled here, in which this is the part of a paternal chromosome, right? This is anaphase. And now what will happen? these are the spindle fibers, right? Now what will happen? Now again the cell will start furrowing from middle. This will start developing nuclear membrane, and these two will get separated, and now they will behave as a single chromosome again, in which one is similar to the maternal chromosome and other is the blend of maternal and paternal chromosome. And after this telophase, I have considered this as one daughter cell. So at last we will be having total 4 daughter cells, right? We will be having total 4 daughter cells. So this is the whole concept of meiotic cell division. Now let us summarize the full process of meiosis with the help of a colored diagrams. By the end of Prophase One, the crossing over has taken place, the centrioles has moved to the opposite poles, and nuclear membrane is fully dissolved. Then let us move to the next phase, that is Metaphase One. In Metaphase One, all the chromosomes gets aligned to the equatorial plane. This equatorial plane the chromosomes has aligned, as well as the microtubules gets attached to the centromeres. Then next phase is Anaphase One. In Anaphase One, the 2 chromosomes starts moving to the opposite poles. But at this stage, we can see that maternal chromosome is not fully maternal. It has few parts of the paternal chromosome, and paternal chromosome is not fully paternal. It has few parts or few genes of maternal chromosome. And also here the centromere is not splitted as in the case of mitosis. Here only the 2 chromosomes, they have moved apart to the opposite poles. Then next phase is Telophase One. In Telophase One, the process is almost just opposite to the Prophase One. That is the nuclear membrane. Here in case of Prophase One, the nuclear membrane has dissolved, but in case of Telophase One, the nuclear membrane reappears again, and the microtubules has dissolved. Centrioles are still there, right? Then the cytokinesis happens. In cytokinesis, this furrow, you can see in telophase there is a cell furrow, and in cytokinesis these are divided into 2 daughter cells. Now the 2 daughter cells, they have nuclear membrane of their own, and then the next stage appears, that is Meiosis II. Now in case of Meiosis II, again all the phases are same, but in Meiosis One, reduction division has taken place. In the prophase we can see there are total number of 4 chromosomes, and by the end of this Meiosis One, we can see one daughter cell has only 2 number of chromosomes. So reduction division has taken place. In Meiosis II, no reduction division takes place, only equational division takes place. The process of Meiosis II is just like mitosis, the process of mitosis. Here, in case of Prophase II, the nuclear membrane starts dissolving, centrioles starts moving to the opposite poles, right? In case of Metaphase II, the chromosomes starts aligning themselves in the equatorial plate. Then in case of Anaphase, the microtubules starts shortening themselves and pulling the chromosomes to the opposite poles. Here the chromosomes are on the opposite poles, right? Then comes the telophase II. In case of telophase II, nuclear membrane reappears, microtubules has dissolved, microtubules has gone back, and the cell has a furrow inside. After this, the cytokinesis will happen again, and in that case, this will give rise to 2 daughter cells, and this will give rise to 2 more daughter cells. So by the end of meiosis process, the full meiosis process, there are 4 daughter cells. One parent cell has given rise to 4 daughter cells, in which the number of chromosomes has reduced to half. So that is all with the meiosis process. Thank you.